Assalamu alaikum. Welcome to the Genomics and Bioinformatics lab. Today we are going to learn how you can make a heat map using the TBTools. TBTools is basically a kind of bioinformatic tools which is used for multiple purposes, and one of them is the heat map generation. For the importance of the heat map is basically used to indicate the interaction between two or more different or same terminologies. It can be also be used to indicate the abiotic stresses like heat, light, and plant presence of plant hormones like indole-acetic acid or abscisic acid, etc. Also, it is used to indicate the tissue expression like certain proteins, how much they are expressed in different parts of plants, and also it can be used for the orthogroup analysis between different species, and also per species orthogroup analysis, etc. Heat map has many other purposes too. So now let's move on how you can generate a heat map. To generate a heat map, you are required to have a data set. And in this Excel sheet, I have two data sets. The one is the for the tissue expression, and the other is one for the abiotic stresses. So for the heat map generation, first you're going to select all this data, copy it using Control+C, and then we are going to open the TBTools so that we can make a heat map there. Okay, so first of all, we are going to the options of graphics, and there's the option heat map illustrator, and click on the heat map. Select all this data, delete it, and now put your data under the set input ID list. Start, and we have heat map here, but this is not the final outlook. You can change multiple settings to have your required heat map. So starting from the beginning, first of all, there is the option of lucky color in which you can change the color of your heat map. Simply click on it and you can see the multiple colors, multiple shades. Okay, so I will let it remain as it is, and then move on the next option, that is the legend font. Legend is this is the legend, and you can change its font, you can change its size, you can make it bold, italic, etc. But no such requirements is no such requirements are needed here. So just let it remain as it is. And there is the option of row name font. Using this option, you can change this area. You can change its size. You can make it bold or italic. But no such requirements are needed here for the heat map I'm going to make. So just let it remain as it is. Then there is the column, and we can change its settings using this option of column name font. So I just want to make it italic, so just click it here and okay. Okay, so now it's in italics. So the next these two options, width and height, these are basically used to adjust the size of the heat map. And in this heat map, the width seems fine. I just want to make its height a little bit less. So yes, this is the right amount of height. And so for next settings, you can click here, show control dialog, and there are multiple options. So there are multiple options here. So there are multiple options here which you can use to customize your heat map. And there is the option of log scale. This option just divides your actual values by 10, and you can see before log scale these were 60, 50, 40, and now they are 6, 5, 4. And there are more options like cluster rows, which will cluster all these rows, and then is cluster columns, and all these columns are clustered. And now if you want to indicate the values of each of the color presented in different boxes, you can simply click on the show value, and here you can see different values are present in each box, indicating that how much the expression of certain genes in different parts of the plant. Next, you can change the value format. Uh, you can see there is the 0.00, and all the options are two decimals. So you can reduce it, let it remain as it is. Next, there are some options like you can change the shape of the boxes. These specific boxes, you can change their shapes. These are in rectangular form right now. You can change them to round rectangular. Okay, so in this case, just let it remain rectangle. Then there are option of switch row and column. I'm not going to select it now. If you want to change these are the rows and these are the columns, and if you want to change their positions, exchange their positions with each other, you can simply use this option. Then root, leaf, and stem are going to be presented here, and all these genes they will be present in the row position. Then further, there are some options. Let me see. For these, you have some options related to the legend. You can set a maximum or minimum value for the legend, and also there is a legend value format. Legend value format is in the 6.00 or 5.00. You can change it to simply 654, but as we have remain our value format is 0.00, so legend value format they should both be same. So we have almost completed all our requirements which are required here to make a heat map. Is we have used the log scale, we have clustered the rows and columns, we have shown the values, and okay, a value format, let it remain unchanged, and also legend value format remains unchanged. It's your choice if you want to if you want to change the tile shape, and okay, or it's all, and now simply toss it, adjust it closer to the heat map, just legend closer to the heat map, and now the how you can save this heat map and then use it in your projects. Save graph, there is the option of .jpg 600 dpi, and then save it. You can select your location where you want to save it, and I would like to save it on desktop. Give it file a name. So issue expression. It's issue expression. then click on Save. This is all. Now you can simply minimize, and here it is in the image form. Now you can insert this image in your Word document. You can adjust it with other images in your PowerPoint, etc. Now this heat map can be easily utilized in your projects, assignments, your research articles, thesis, etc. So that is all from my side. Thank you very much.